Okay, B3, factors affecting the activity of enzymes. So we've gone through the structure of enzymes. We've gone and talked about models of enzyme activity, right? So is this shape, this active site, these fields that combine to catalyze reactions, have things happen. Our body usually is breaking covalent bonds or stitching covalent bonds together. The key here, enzymes lowering the activation energy for those reactions to happen. Now, how is this controlled? How is enzymes? How is the activity of these enzymes controlled? Right. So there's something called homeostasis. You may have heard the word homeostasis. What homeostasis is basically it's the conditions under which enzymes work best. So your cells are able to function and carry out metabolism, and you're able to live. So your body is always trying to maintain homeostasis. It's this baseline. We're human beings, we're 37.5 degrees centigrade, something like that. So that is part of homeostasis. Humans we're endothermic, which means we control our body temperature, because temperature is one of the things that affects enzyme activity. We'll talk about that in a second. The other thing is pH. So when we're dealing with biological systems, pH and temperature are very important. Remember, we talked about pH. If you forgot about pH, go look in A1. We talk about pH, right? pH is the concentration of hydrogen ions or hydroxide ions, the ratio of hydrogen ions to hydroxide ions in a solution. Humans are a solution. We're a solution, mostly water. We're a solution of water and stuff in the water. So there are optimum conditions under which enzymes work. So optimum temperatures under which enzymes work, and optimum pH under which enzymes work. And those optimum conditions are essentially controlled by that sequence of amino acids and the chemical properties of the amino acids, and basically those electrochemical properties of amino acids. So how does temperature affect enzyme activity? Well, remember we talked about enzymes, how enzymes work, right? That they lower the activation energy of chemical reactions, right? So you have reactants, you have a substrate, you have reactants. These are going to produce a product. And when it's really cold, when it's super cold, temperature, remember, is just kinetic energy. Thermal energy is just kinetic energy. It's how fast molecules are moving. And when it's cold, these molecules aren't moving very fast, and they are not moving with a lot of energy. When they bump into each other, not a lot happens. The same thing happens when you have an enzyme in that equation as well, because now it's cold. This enzyme is a molecule in this system, and it is moving slowly, very slowly, and so it will take a long time for it to encounter its substrate. And also when it encounters its substrate, because things are kind of rigidy, it might not exactly bind because things are cold, so it might be harder for it to bind. You need a little bit of energy, right? You still need activation energy in an enzyme catalyzed reaction. You need a little bit of energy for the enzyme to perform the reaction and merge these two things together, let's say. So reaction rate, when things are colder, the reaction rate is going to slow down because the substrate, the enzyme, everything is moving slower and not with a lot of energy. So these might be bouncing into each other, bouncing, and then finally it makes it in, and these things break off, and the enzyme continues on. As you increase temperature, now things are moving around faster. The likelihood that this enzyme is going to encounter its substrate is a lot more, and it's going to encounter another one again, and so the reaction rate as temperature goes up increases. Except sometimes it does not, because there's an optimum range of temperature for enzymes to work, because eventually what can happen is now things are moving really fast. They're slamming into each other. And remember that three-dimensional shape of enzymes, that tertiary structure and quaternary structure, which is different peptide chains coming together, is often held together by hydrogen bonds, which are weak bonds. Those hydrogen bonds, those regions of positive and negative fields that are creating those hydrogen bonds, are weak bonds. And if you have temperature, all the molecules, all the atoms in the individual atoms in the enzyme are starting to vibrate and move, and the enzyme is starting to vibrate and move, because temperature is not only the molecules moving faster, but also them vibrating faster. And eventually what can happen if temperature increases too high, that vibrating becomes too great and the hydrogen bonds can't hang on. And that is what we call the enzymes denaturing. The enzymes will denature. They will lose their shape. And the active site stops being the active site anymore. It loses its shape. The enzyme can't do its job. So if you were to graph temperature, it might be something like let's say we have a graph here. And so this is going to be temp. And this is reaction rate. And so as you increase temperature, you might have the temperature going up, the reaction rate is going up, but then it reaches a point where the enzyme denatures and reaction rate goes to 0 because the enzymes are broken, they're denatured. Now, you might be able to lower the temperature, and those hydrogen bonds might actually reform back. As the energy lowers, the hydrogen bonds might reform back, and the enzyme might start working again. Certain enzymes that might work. If you increase the temperature too much and those covalent bonds in the enzyme and the ionic bonds that are holding tertiary structure together break, not likely that you're going to get, the enzyme won't work again. You may break it too much. The same thing, some enzymes need guides. They need other enzymes when they're being assembled to make sure that the hydrogen bonds and things form in the right place. Those enzymes also will not return. But it is possible, once you lower the temperature, that the hydrogen bonds reform and the enzyme can work again. Fever. That's why you get fevers when you're sick. You get a fever when you're sick because what your body is trying to do is to denature the enzymes of your attacker, the bacteria that's attacking you or the virus that's attacking you. Viruses and bacteria, their optimum range needs to be cooler than our body. Our body is at the hotter end of their optimum range because they need to exist outside of the body in the real world. So their enzymes need to be in such a way that they can operate at cooler temperatures as well as the hotter temperatures. But there's just so much that you can do with an enzyme, so that range is limited. So our body, our normal body temperature, is at the upper end of their operating range. So if we start to get hot, we start to push out of their regular operating range. Our enzymes can still function. Our enzymes, because they're made to work in our body, their temperature range is a little bit greater. Our enzymes work a little bit hotter, so it won't hurt our enzymes, but will denature the enzymes of viruses and bacteria. Now, obviously, our enzymes get denatured too. You can die from fever. That's why you die from fever, because our body sometimes it gets so excited about killing viruses and bacteria that it kills us too. So that's optimum temperature. pH. pH is the other thing that can affect temperature. Why does pH affect temperature? Well, think about it. Remember, enzymes are these fields. They're held together by these hydrogen bonds, which are fields of positive and negative space. pH is the ratio between hydrogen ions, which we talked about. I think we talked about in class. Hydrogen ions are essentially protons, positively charged protons, and hydroxyl groups, which are hydroxide groups, which are these negatively charged oxygen and hydrogen complexes. So if you, let's say, lower the pH of something, make it very acidic, that solution that enzyme is in becomes very acidic, which means it's got a whole bunch of hydrogen ions, of protons everywhere, positive fields everywhere, and those are going to start affecting the hydrogen bonds in the enzyme, because they're like attracting to those bonds, pulling on those bonds, and the enzyme will lose its shape and again could denature if things become too acidic. And that all depends on the nature also of the R groups, the amino acid R groups that are what are their properties in the active site or around the enzyme. They could be negatively charged, so as they attract protons. They could be positively charged, they repel protons. The same thing with if a substrate is too alkaline, it's got too many hydroxides. Those are negatively charged. Those electric fields will also pull on the hydrogen bonds and can denature and change the shape of the enzyme if there's too many of them. So there's a range of pH that enzymes work in before the hydrogen bonds that are keeping that tertiary structure of the enzyme together become effective. So if you draw reaction rate, so not temperature, but you draw oh, you know what? My temperature, I just realized my temperature reaction rate drawing was totally wrong. I labeled them wrong. So this should be temp, and this should be reaction rate. So let's just do that again. So reaction rate, temperature. As temperature increases, reaction rate increases until temperature and reaction rate drops down to 0. That makes sense when the enzyme gets denatured. Now, pH. So if we change this, to pH, what you're going to see is the graph is going to look like this. So there will be an optimum pH where the enzyme works best. Can I zoom in on that? Is that a possibility? No, can't zoom in this mode. There's an optimum pH where the reaction rate is highest, and then as you become more acidic, it drops off, and as you become more alkaline, it drops off. Okay? Enzymes can have the same substrate, but they can be different enzymes and work in different pHs. So when we look at the digestive system, there's trypsin and pepsin. Pepsin works in your stomach, which is super acidic, to break down peptide bonds. You look up what peptide bonds are. And trypsin works in your duodenum or duodenum, which is more basic, to do the same thing. But the different enzymes, so they have different optimum pHs. Okay, so that's optimum conditions: temperature, pH. Now, other things that affect enzyme reaction rate are inhibitors. So there are two types of inhibitors. Three types of inhibitors. So the first type is called a competitive inhibitor. Okay, so let me show you how a competitive inhibitor would work. This might work. Okay, so let's say these are my, I love these things. These highlighters are my substrates, okay? These highlighters are my substrates, here, the highlighter is, this complex is a highlighter. It fits into this active site, which uses induced fit. It breaks the two, and this is a catabolic reaction, probably exothermic, hydrolysis of breaking a polymer or a disaccharide into monosaccharides, or polymer into individual components. Okay, so what does an inhibitor do? Well, imagine, look, this isn't a highlighter, but it's a dry erase marker. It's similar, but slightly different. And it could be part of something like this. Let's say, so this is different. It could be this, it looks the same, but it's slightly different. And so it might fit into the active site of this enzyme, which breaks down highlighters, but it's slightly different. So now maybe the enzyme can't break this bond. This bond has different properties. It's stronger, so whatever the enzyme does, so this keeps coming in and stuck. It's blocking the active site. And now the thing that we want to do can't happen. It's being inhibited because this is competing with our highlighter. That's what a competitive inhibitor does. Is a competitive inhibitor fits into the active site and doesn't let the enzyme carry out the reaction that it wants to carry out, because it's blocking the active site. And it might not, this might be part of some huge thing, it's some big thing, and that fits into the enzyme, and there's no way it can break it, but it's just a piece of it looks similar enough that it blocks the active site. That's what a competitive inhibitor does. Okay? Is there anything else I want to say? Yeah. Well, I guess a good example is antibiotics. That we use on bacteria are often competitive inhibitors, and they will block the active site of some enzyme that the bacteria uses to assemble some part of its structure. So like penicillin blocks is a competitive inhibitor for an enzyme that assembles cell walls in bacteria. And so it blocks the ability of the bacteria to build its cell wall, and therefore the bacteria can't grow, can't reproduce, can't survive. Okay, now non-competitive inhibitors. What non-competitive inhibitors do is basically, so here's my enzyme again. It forms the substrate enzyme complex, breaks this. These guys go floating off. The enzyme stays behind. These guys go floating off. What a non-competitive inhibitor does is it doesn't go into the active site. What it does, what would be a good example of a how about this? What it does is, let's say it binds here. Ow! It binds to the enzyme somewhere. Ow! Somewhere not in the active site, somewhere else. Ow! And so now it's changed the distribution, those fields, those electron fields, and now the enzyme can't move, or the active site is distorted somehow. And so now when the substrate comes in, the enzyme can't do its job. It can't break this because it's been changed, or the active site is changed so much that the substrate can't enter. So a non-competitive inhibitor just binds not at the active site of the enzyme and just changes the electron distribution and the shape of the enzyme so that the active site is altered and the enzyme can't do its job. Heavy metal ions are non-competitive inhibitors. Remember we talked about so heavy metal ions are going to be like lead and what's a good example? Don't eat lead paint. Just big heavy metal, like uranium is toxic. It's toxic for other reasons, because it's radioactive, but it also would be toxic because it's a huge, if you had uranium ions in your body, they would be not only radioactive, they would also distort the shape of your enzymes, because that positive charge of that huge metal ion will, as it comes close to the enzyme, will pull on the hydrogen bonds and distort the active site and not let the enzyme function. Okay, boom. That's all I need to say. That's all I need to say about inhibitors. That is very straightforward. Okay, so the last thing is substrate enzyme concentrations. Okay, this one is getting long. This is a little longer subject. So substrate enzyme concentrations, what do you need to know about that? Well, okay, so pH, reaction rate. So if I've got endless substrate and I add enzyme, the reaction rate as I add, so here we go, we're going to say number of enzymes. So enzyme concentration, enzyme, this is the symbol for concentration, enzyme concentration, okay? So as enzyme concentration increases, there are more and more and more active sites to carry out the reaction. So the reaction rate is going to go up as enzyme concentration increases. As long as there is substrate, the reaction rate will go up because there are active sites to carry out the reaction, to be a catalyst for the reaction. Now, That makes sense. So you want to increase the reaction rate, increase the number of enzymes. Enzyme concentration. How does substrate concentration affect the reaction rate? So here we have substrate concentration. Okay, substrate concentration. Can you guys see it? I don't know if you guys can see it very well, but you can see it well. Okay, what's going to happen with substrate concentration is you've got a whole bunch of enzymes, and now substrate comes in. And the more and more substrate comes in, the more and more it encounters those active sites of those enzymes, and the reaction rate goes up and up and up. But at a certain point, you're increasing substrate, you're not increasing enzymes. All the active sites on all the enzyme molecules will be saturated with substrate. So if you imagine, I lost that. So all the active sites will have, there's that moment where they all have a substrate in them, and they make a product and grab the next substrate and grab a product. But if there's more substrate than there are active sites, you will reach a maximum, basically, is what happens. You reach a maximum. Hold on, let me grab that pen. So when you reach the maximum, when all the enzyme active sites are saturated, they're all performing now, they're all catalyzing, being a catalyst for the reaction rate levels off and plateaus. Unless you increase, there's just a maximum. Unless you increase the number of enzymes, you will reach a maximum if you're increasing substrate concentration. Okay, that's it for this. So we talked about the ways that you can affect activity of enzymes. So temperature, pH, competitive inhibitors, non-competitive inhibitors, heavy metal ions, substrate concentration, enzyme concentration. And your body manipulates all of those things. We like heavy metal ions. We try not to have heavy metal ions. And also, I guess those cofactors are another way of affecting activity of enzymes. You need cofactors, so vitamins and metal ions, but not heavy metal ions like zinc and potassium, to make enzymes work as well. Those would be another one that is related to this. Okay.